Hey guys, what's up? Okay, today's video we will see a lot of things about tissue engineering and regeneration. Of note, tissue engineering is a really recent and enormously growing branch. It is a very incredible branch in that we will do awesome things like heart transplant using a heart which we actually make in the laboratories. Also, guys, we will be able to produce other organs as well in tissue engineering. That is what tissue engineering is all about. So we will see the various methods how by which this is done. Let's get started. So tissue engineering is a new and expanding field. The basic concept is that if the bone is able to form a lot of hematopoietic cells, the cartilage is not able to form it. Let's be the bone and form the entire human organs and to grow an organism. So this tissue, which is going to be used in tissue engineering, the hero of the tissue engineering concept, is grown either in the laboratory or inside the body of the human being itself, called in vitro and in vivo. There are various opportunities, and the most important one is called the stem cell therapy. Stem cell therapy, the stem cells are cells which can virtually multiply to form any cell in the human body. It can be a skin, a blood vessel, a heart, a bladder, pancreas, GIT, anything, which means these are basically the new alternatives to lab rats, right? Yeah, true. Applications include we can test any sort of a treatment uh, by using these things as the model. We can also use these for the treatment, for example, as in for liver transplantation, or we can do surgical assessment using these models as well. The key areas is the long-term goal of the tissue engineering process is just we have to replace the entire organ of the human being. So where do we get the we can't just build organs from dirt, right? So we need some cells after which we can form new cells. So there are two sources, the somatic cells and the stem cells. These are the various types of the stem cells, such as the somatic stem cells. These are not the same as the somatic cells. They are different. They are somatic stem cells. We'll see more about them. Mesenchymal, fetal, embryonic, induced pluripotent stem cells. Make yourself familiar with these terms, guys. Pause this video and you can revise it till now. So about the somatic cells are nothing but differentiated specialized cells in contrast to stem cells. Stem cells are undifferentiated and non-specialized cells, but somatic cells are differentiated and specialized. For example, a skin is a somatic cell, which means it will multiply to form the host which is lost. For example, if there is an aberration and then the skin grows in order to replace what is already gone, means a skin will differentiate only to a skin. It doesn't differentiate into a bone. That's what they mean by it is differentiate and is specialized cells, which is very good. So it can be of two types when we use somatic cells. It can be autologous or allogenic, meaning we use the same person's cell to form the new cells. So whereas allogenic means the same species is used but not technically the same person. So autologous is always beneficial in that there is no risk of rejection and no need to do immunosuppressant. However, the limitation is that it does not have indefinite replication potential. Moving on to the stem cells, which are circumventing that drawback of the previously read somatic cell, that it has unlimited replication potential. And as always, guys, undifferentiated non-specialized cells or somatic cells. That is, A can give rise to multiple A, that is hundreds of A, suggesting there's unlimited replication, or A cell can become B, C, D, E, F to suggest that it can undergo any specialization, whatever it wants. So stem cells can be somatic, mesenchymal, induced pluripotent stem cells, fetal and embryonic stem cells. So guys, first you need to familiarize with the terms. There are three obvious kinds of stem cells, which are multipotent, pluripotent, totipotent. Well, these terms can be a bit confusing, but let me make it clear. Multipotent stem cells or stem cells only. Multipotent stem cells are going to multiply, but it will multiply only to a certain extent. It will multiply a lot, but only to a certain extent. For example, if it's in an endodermal multipotent cell, it will form all tissues in the endoderm alone. But if it's a mesodermal, it forms all tissues only in the mesoderm. Okay, that is about the multipotent. But if it is a pluripotent stem cell, it can form literally any cell in the entire adult human being. But what a totipotent stem cell means, totally anything it can form. For example, it can form an embryo embryonal cell or a fetal cell or an actual adult cell. So this is the main difference between multi, toti, and pluripotent cells. Okay, so this we'll go into the adult one. Adult tissue resident or the somatic stem cell. It's a multipotent stem cell because it has the ability to differentiate into multiple cell types, but not all the cell types. Examples include those in the GI mucosa and in the bones, that is the bone marrow. Second, there are these types of somatic stem cells, such as the hematopoietic, those blood-forming ones, endothelial progenitor, the neural, mesenchymal, or the stromal ones too. Let's go to the bone. So mesenchymal stem cells and the stromal cells. The source is the bone marrow or the adipose tissue and the umbilical cord. The cluster of differentiation which is present at the cell marker include 73, 90, and 105, whereas which ones are absent are very important. 34 and 45 CDs are absent. Well, these cells can produce growth factors, can also specialize into either of these things, either osteoblast, chondrocyte, adipocyte, tenocyte, myocyte, anything, because they are derived from the bone marrow, adipose tissue, and the umbilical cord. And let's move on to the other one, which is embryonic stem cell, which are totipotent, meaning embryonal, fetal, and the entire adult cells are also going to be formed from this, meaning all types of cells in short. The source is the inner cell mass of the early blastocyte. Just 4 to 5 days after fertilization, we are going to take it. But however, ethically, it is having a really bad drawback that ethical considerations are there. Immune rejection could also take place, but however, we can circumvent that by saying, so there is this oocyte. We can replace a nucleus of this oocyte with the recipient's nucleus. Means technically, it's going to be the auto auto stem cells, right? Not allo stem cells. They are auto. Means that's how pluripotent stem cells are formed, guys. We take a cell which has a lot of replication potential, that is the oocyte, and the recipient nucleus is taken and pluripotent stem cell swarmed, and our goal is achieved. Moving on to the fetus. The fetal stem cells are pluripotent, and they are also tried to be used in Parkinson's disease, diabetes, spinal cord injury. They are similar to embryonal stem cell, where ethical issues are the main drawback. Induced pluripotent stem cells are yet another ones. These are pluripotent. The source is a specialized somatic cell. Woo. Okay, so what happens is that the somatic cell is added with a virus. This virus has transcription factors which will be transported into the somatic cell's nucleus, which is the process is called the transfection, which occurs in vitro, means in the laboratory, to form the induced pluripotent stem cell. So basically, induced pluripotent stem cell means it's an addition of a virus to a somatic cell. Well, sometimes you can also store all these kind of cells in a bank and use it whenever necessary. So these somatic cells can be replaced by a somatic cell by a donor and stored in the bank and used alternatively after doing cross matching. However, the drawback is that a virus is used. This virus is usually the viruses which are usually used are retroviruses and lentiviruses. These viruses have oncogenes. They will have the potential to stimulate oncogenes. We don't want to give the patient tumor, which is why we use viruses which do not cause cancer, such as adenovirus and Sundai virus vectors. But what if we don't want a virus at all? We have a solution for that too. We can use molecules or growth factors and chemicals in place of viruses. They can also do the same job as the viruses do. So in vitro differentiation of the stem cells to specialized tissues. So this process is that the stem cells are collected in a petri dish with the culture media, and then it is used. So basically it's like a cooking process. It's like making a tea. We can make tea two ways. One, we put the powder and then boil the milk. Two, we boil the milk and then put the powder. So the main thing over here which I want to emphasize is that the ingredient is very important, but the preparatory method is important too. So the stem cell A, if the stem cells are added with ingredient A, it's going to become skin. Just like that, guys, milk is the stem cell here. A can be coffee powder, tea powder, or a badam drink. You can add anything. It forms anything. The fact is that there are some factors which will decide the outcome, such as the biomaterials which are used, the attribute of the scaffold, and mechanical stress, whatever is given. Okay, so what is a scaffold, guys? A scaffold is nothing but a brick in a building. I would consider it as a brick in the building, or a sponge which has a lot of pores onto which water molecules can sit. So this scaffold is going to be the building block of our entire organ. So our aim is to mirror the in vivo development. We either use embryonal or induced pluripotent stem cells. First step is to convert it into either ecto, endo, or mesodermal line, and after that it can specialize into whatever cell is desired. So the bricks are nothing but scaffolds over here. The scaffolds are the bricks of a home, and okay, so that's the analogy here, but the actual chemical nature of scaffold is it's an extracellular matrix mainly made up of collagen. Usually cell cultures are done, they are done routinely. These cell cultures are going to form cells which are in the form of sheaths, right? These sheaths are usually called organoids, but what our aims is to form an entire organ. Sheaths don't match an organ, right? Which is why we need the cell culture plus the scaffold. The cell culture will give us the cells. The scaffold will give us the base bone, the base for the organ. So it gives the physical support, the shape, and also cell signals are also given by, like the growth factors are given by the scaffold. These signals, which are given by the scaffold, will ensure growth, migration, as well as differentiation and multiplication of the cells. There are two types of scaffolds. Both of them are obviously three-dimensional because our organs are three-dimensional. They are rigid or semi-rigid. The source of this scaffold, it can be either a natural one or an artificial one. If it's a natural one, it's a human tissue. Artificial, it's made up of a lot of biomaterials. However, there are some general features which are the characteristic features which are good for a scaffold. One, it has to have a lot of pores. Two is it has to allow cells in order to grow, multiply, and divide as well, and differentiate as well. Three, it has to have pores, right? So it has pores. It has to allow raw materials like oxygen, nutrients, everything to enter into the cells and nourish them. Four, it has to deliver the signal such as growth factors. Five, it should be non-immunogenic. Six, biodegradable, biocompatible, bioreabsorbable. First is the natural scaffold. Natural scaffold is a human tissue. An entire organ is needed for this purpose. So an entire liver or an entire kidney can be taken as an example. So these organs are taken along with your intact blood vessels. So what exactly we do is that inside the blood vessels we pour detergent in order to kill the cells which are already present there. So that what is left over is only the extracellular matrix, which is technically the scaffold. This process can also be done alternatively by simply immersing the entire organ within a solution of detergent. Our entire aim is to repopulate the the scaffold by adding new cells after replication by the cell culture techniques. However, autologous cells can also be incorporated, which is also possible, but however, which is the limiting aspect of this natural scaffolding is that an entire organ supply is pretty limited. Well, about the artificial ones, it can be either natural, artificial ones, or synthetic ones. Natural as in it's going to use collagen, cellulose, gelatin, fibrin, or polysaccharides which are available naturally, or it can be synthetic such as uses polymers, ceramics, and glasses. Polymers which are used are graphene, polyglycolide, and polylactide. Ceramics Ceramics are rich in calcium hydroxyapatite, which is a rich component in the bone. Well, this is a 3D printer, whereas this one is a it's an electro-spinning device. It's called an electrospinner. So the technologies which are used to do this tissue engineering include 3D printers, which predominantly use a gel. It's called a hydrogel. We'll know more about the hydrogel. So about the hydrogel is a thing which will be able to attract a lot of aqueous moieties to it. That is the feature which I want you to remember. Hydrogel attracts aqueous. Hydro means something with the water, so it attracts aqueous a lot. Electrospinning. In electrospinning, either the polymers alone will spin or polymers and the ceramics are used together, just like as in the polymers as in graphene mainly used, ceramics such as hydroxyapatite, calcium hydroxyapatite. So either polymer is used alone or polymers and ceramics are used together. Well, if both these things are used together, both advantages are coupled, which is why we call it the smart electrospinning technique. However, there is a disadvantage that we won't be able to incorporate the blood vessels, but no worries, we've got 3D printers and VEGF on our way to our help. Next is the approaches to cell seeding of the scaffolds and the bioreactors. This is again an in vitro technique, which means it is done inside a laboratory. The aim of this is to induce a rapid seeding. It has to be really fast, get big fast, high efficiency, and there should be uniform penetration. Meaning, say we have 3 points in a scaffold A, B, and C. If point A receives 100 cells, we will expect point B and point C to receive 100 cells as well. But if it is non-uniform, A will receive 100 cells, B will receive 1,000, C will receive 10 cells, which is undesirable. So these three are the aims. So there are 6 techniques about how we do it. So this one, for illustration purpose, this can be considered as the cells which are going to be added to this scaffold. Okay, so static means we basically place the scaffold in a plate, and after that we place these cells, assuming that they will get incorporated, assuming that the cells will get incorporated into the scaffold by themselves passively. But however, rapidness is not possible here. It's not even high efficiency. Uniform penetration can be there, but two of their aims is not satisfied, which means we need another technique. It's called the dynamic technique, where rotation is the main key. It is going to form a centrifugal force. So we put the culture media and the cells in a beaker, and we are going to put the scaffold inside the same beaker, and we are going to rotate only the scaffold. Alternatively, both the scaffold and the culture media could be rotated, forming a centrifugal force. So it is going to be of high efficiency and also rapid feeding, along with uniform distribution. Magnetic ones could also be done. Magnets, so the cells are labeled with the supramagnetic microbeads, and the scaffold is coated with magnets, so those two will attract each other, and they will be very good. But however, there's a disadvantage that it can become toxic or something. And fourth method is photopolymerized hydrogel. A photopolymerized hydrogel is very good because we already know what hydrogel is. Hydra attracts aqueous. So what it does is it has a property of attracting a lot of fluid, or it will absorb a lot of aqueous when it retains shape. For example, if you think about a sponge will retain the shape even if it has a lot of water. So a hydrogel is somewhat similar to a sponge, which is mainly used in 3D printers technology. So what exactly we do in this is that we take a medium, and monomer cells are placed inside, and we are going to coat the hydrogel over here in the scaffold. It will very well attract it, but in order to make it more efficient, we are going to add ultraviolet rays. Ultraviolet rays will polymerize these monomers, which will attract even further. And the next one is that we can alternatively just not only UV rays, we can also use an adhesive tape. Just like when our paper is broken, paper is torn, we use an adhesive sellotape. Here, because in molecular level we use a peptide, it's called an adhesive peptide made up of AGA. AGA stands for arginine, glycine, aspartic acid. So these three things in a form of a peptide is a very good adhesive agent. Differential pressure is the fifth technique, otherwise called a vacuum seeding. Vacuum is somewhat similar to osmosis, except that your active pressure, that is active vacuum, is given. It's not advantageous. I mean, it's advantageous in one way, that is, it is rapid, but the viability of the tissue might be reduced by this technique because it's like strangulating a cell. And the last one, which is the most important one and the most complex one of all the above, is the bioreactor perfusion. Bioreactor perfusion, as always, guys. So over here, there's this blood vessel. Initially, we poured a detergent inside this blood vessel, but now we are going to pour cells in the decellularized scaffold. Okay, we are going to we first pour the detergent to decellularize it, but now it is already decellularized, so we want to cellularize it, which means we pour the cells. That's the basic concept there. However, after doing all these, it's not yet successful. There are a lot of challenges to this in vitro technique. First of all, it's like adopting a child. So you have to meet all the requirements. You have to give the oxygen, the food, such as the nutrients, the growth factors, that is, you have to make it go to the school. So it has to meet all the requirements. Second is that the number of cells have to be adequate. Whether it is viable or not is essential, because we don't want dead cells to be inside a body of the patient, right? And it has to be functional, and most important of all, it should never revert back to a normal, to the pre-state. I mean, it should never revert back to the dead state. And most importantly, that if this particular group of stem cell tissue, which is needed for a particular function, as in it is going to be a part of a skeletal muscle or a cardiac muscle, it should be in a way that it is capable of maintaining and holding its shit together, because it shouldn't break up itself when it is having a lot of pressure, as in the cardiac muscle is supposed to pump blood to the entire body. So implantation is the main key, meaning we have to restore the functional integration of the particular tissue we are dealing with, for which we have an innovative high resolution 3D printing technology which is available. Functional integration is the success of the entire tissue engineering technique. So there are certain safety concerns, as in these stem cells are going to develop tumors and they are going to cause infection. Tumors are mainly due to virus, so we can remove the virus. Infection, you can screen the person who is the donor for it, so infection is not going to be a problem. And third is epigenetic and genetic. Again, you can screen for genetic diseases in the person who is the donor. And fourth, this is the tissue viability or the function is reduced. It's basically on our fault if this happens, because the viability and functional maintenance is what we are appointed for. And then differentiated to undesired cell. For example, if we wanted a hard muscle, if a smooth muscle comes, it's of no use. And last two things are due to this allogenic concept, meaning rejection and immunosuppression side effects, which will not occur if autologous graft transfer occurs. In the future, this is a very dominating and a rapidly growing field, especially the main point is that it is commercialized. So yeah, stock market, real estate, come on, rise here. Well, as always guys, read Bailey and love. Thank you. See you soon.